Welcome, curious minds, to another deep dive. Today, we're going on quite an incredible journey. It starts with those intricate blueprints stored in our DNA,
Mhm.
and it ends with the actual functional machinery that makes life possible. Proteins. I mean, think about it. How does that silent code in our genes actually become, the stuff that does everything in our cells?
It's a fantastic question, and it really gets to the heart of molecular biology. So in this deep dive, our mission is to unpack exactly that, the process of translation. We're drawing from Chapter 13 of Essentials of Genetics.
Okay.
We'll look at how messenger RNA, mRNA, gets decoded to build polypeptide chains, and crucially, how those chains then fold into the proteins essential for, well, everything.
And hopefully what you'll gain is a really clear picture of the key molecules involved, the almost choreographed steps, and even some of the big historical moments that showed us just how central proteins are. Get ready for some aha moments. Okay, let's dive in.
All right, so just to set the stage quickly, remember from our last discussion, the central dogma starts with DNA storing genetic information.
Right.
That info is then transcribed into messenger RNA. Think of mRNA as the working copy.
Got it. The DNA stays safe, the mRNA carries the message out. So what's next? How do we get from that message to the actual functional protein?
That is precisely where translation comes in. It's essentially the biological process of polymerizing amino acids into polypeptide chains. We're translating literally from the language of nucleic acids, the mRNA sequence, into the language of proteins.
Okay, so the mRNA sequence tells the cell exactly which amino acids to string together and in what order.
Exactly. The ribonucleotide sequence in the mRNA dictates the amino acid sequence of the protein. It's a direct code.
And this transformation, it doesn't happen in a vacuum, right? What are the main molecular players here?
You're right, it's a team effort. Three essential things interact.
Yeah.
The mRNA itself, carrying the instructions, transfer RNA or tRNA, which acts as the crucial interpreter, and the ribosomes, which are the cellular factories where it all happens.
Ah, the ribosomes and tRNA. I remember Francis Crick had a key idea about tRNA back in the 50s.
He did indeed. In 1957, Crick proposed the brilliant adaptor hypothesis. He figured there had to be a molecule that could bridge the gap between the mRNA code and the amino acids.
Makes sense.
He suggested tRNA molecules were these adapters. Each tRNA recognizes a specific three-nucleotide sequence on the mRNA that's a codon and brings the corresponding amino acid. They have an anticodon end that pairs with the mRNA codon and another end that attaches to the specific amino acid. It's like a molecular Rosetta Stone.
So the tRNAs are the delivery trucks, bringing the right building blocks, and the ribosome is the construction site. Tell us more about these factories.
Precisely. Ribosomes are the protein synthesis factories. They're the universal workbenches where any protein can be assembled based on the mRNA instructions. Structurally, they're made of two subunits, one large, one small.
Okay.
And both subunits are themselves complex assemblies of ribosomal RNA, rRNA, and numerous ribosomal proteins.
And they come in different sizes, right? Like 70S in bacteria and 80S in eukaryotes. What does the S mean again?
Right, different sizes. The S stands for Svedberg unit, which relates to how fast they sediment in a centrifuge. It's a measure influenced by mass, density, and shape. So importantly, these S values aren't simply additive. You can't just add the S values of the subunits to get the S value of the whole ribosome.
Ah, okay. Density and shape matter too. Got it.
Yes.
So when scientists finally got a really good look at these ribosomes using techniques like X-ray diffraction, what was the big surprise? We used to think proteins did all the catalytic work.
That was one of the most stunning revelations, actually. For a long time, yeah, the assumption was proteins did the heavy lifting. But the high-resolution structures, thanks to work by Ramakrishnan, Noller, and others, showed something incredible.
What was it?
The interface between the large and small subunits, the very place where the peptide bonds between amino acids are formed, is made almost entirely of RNA,
Wow. Not protein.
not protein doing the key chemical reaction. This confirmed that the rRNA itself performs the crucial catalytic function. It acts as a ribozyme, an RNA enzyme. It fundamentally changed our view of biological catalysis.
That is fascinating. So RNA isn't just carrying the message, it's actively building the protein chain. What else did these detailed views reveal?
Well, they confirmed that the key sites in the ribosome, the A, P, and E sites, where tRNAs bind, make direct contact with the tRNAs via rRNA. They also showed the ribosome is incredibly dynamic, changing shape as it works.
Dynamic, how so?
And then there was a really surprising finding around 2010 from Niels Fischer's group using cryo-EM. They found the ribosome seems to harness Brownian motion, the random jiggling of molecules due to thermal energy.
Random jiggling, not like a little motor.
Exactly. It suggests some of the conformational changes needed for the ribosome to move along the mRNA are inherent to the ribosome's structure, driven partly by this thermal energy. It's less like a deterministic machine and more like a very sophisticated molecular ratchet using ambient energy.
That is truly mind-bending. A tiny machine powered by random bumping. Okay, let's zoom in on those critical adapters, the tRNAs. What are their key features?
Well, first, they're relatively small molecules, usually 75 to 90 nucleotides. They're also very stable, and their fundamental structure is conserved across bacteria and eukaryotes. And when Robert Hawley sequenced the first tRNA back in 1965, he found something unusual: modified bases. These aren't the standard A, U, G, C nucleotides.
Modified bases, what do they do?
They seem to be important for the tRNA's stability and its precise three-dimensional shape, helping with things like hydrogen bonding. Hawley's work led to the classic two-dimensional cloverleaf model.
Right, the cloverleaf. I remember seeing diagrams of that. How does that help us understand its function?
It clearly shows the key parts. The anticodon loop at one end, which reads the mRNA codon, and the 3' acceptor stem at the other end where the amino acid gets attached.
Okay.
But the real shape, revealed later by X-ray crystallography by Alexander Rich and others, is actually an L shape in 3D.
An L shape.
Yeah. And this L shape cleverly positions the anticodon at one tip and the amino acid attachment site at the other, perfect for its role as an adapter.
Before it can do its job, though, the tRNA needs the right amino acid attached. This charging process sounds absolutely crucial for accuracy. How does that work?
It is absolutely critical. This activation, or charging, is done by a set of enzymes called Aminoacyl-tRNA synthetases. There are 20 different ones, one for each type of amino acid, and they are incredibly specific.
20 specific enzymes. Wow.
Yes. Each one recognizes only one type of amino acid and only the tRNAs that are supposed to carry that amino acid. The enzyme first uses ATP to activate the amino acid, then attaches it to the correct tRNA's 3' end with a high-energy bond. Get this step wrong, and the wrong amino acid goes into the protein.
And how do we know it's the tRNA, not the amino acid itself, that the ribosome recognizes? Was there an experiment that proved this?
Yes, a truly elegant experiment by Chapeville and colleagues in 1962. It definitively proved the tRNA is the adapter.
What do they do?
Okay, so they took tRNA specific for the amino acid cysteine, charged it with radioactive cysteine, tRNA cicity. Then, and this is the clever part, they chemically converted the attached cysteine into alanine while it was still attached to the original tRNA cis. So now they had alanine attached to a tRNA that should code for cysteine.
Okay. So the amino acid is alanine, but the tRNA thinks it's cysteine.
Exactly. Then they put this modified tRNA into a cell-free system with an mRNA sequence that's specifically called for cysteine codons. And what happened?
Let me guess. Alanine got put in.
Precisely. Alanine was incorporated into the polypeptide where cysteine should have been. This showed without a doubt that the ribosome reads the tRNA's anticodon, not the amino acid it happens to be carrying. A landmark experiment.
That really nails it down. Okay, so we have the players: mRNA, charged tRNAs, and the ribosome. Let's get into the actual molecular dance of translation itself. What are the main stages?
It's a continuous process, but we usually break it down into 3 main phases.
Yeah.
Initiation, elongation, and termination.
Initiation, elongation, termination. Got it.
We can focus on the bacterial process first, as it's a bit simpler, and then touch on eukaryotic differences. And remember those 3 key sites on the ribosome we mentioned: the A aminoacyl site, the P peptidyl site, and the E exit site. These are crucial docking spots for the tRNAs.
Okay, so how does it all kick off? What happens during initiation in bacteria?
Initiation in bacteria requires the small ribosomal subunit, the mRNA molecule, a special initiator tRNA carrying N-formyl methionine, or FMET.
FMET, not regular methionine.
Right. In bacteria, the very first amino acid is typically this modified form, FMET. The start codon on the mRNA is almost always AUG, which signals for this FMET. You also need energy from GTP and several protein initiation factors, or IFs.
And how does the ribosome find the right AUG codon to start at? mRNAs are long.
Good question. Bacterial mRNAs have a specific sequence just upstream of the start AUG called the shine-dalgarno sequence. This sequence base pairs with a complementary sequence in the 16S RNA of the small ribosomal subunit.
Ah, so it lines things up perfectly.
Exactly. It positions the small subunit correctly over the start codon. Then the initiation factors help bring in the fmed-tRNA to the P site, not the A site, the P site for the first one, setting the reading frame. Finally, the large subunit joins, GTP is hydrolyzed, the IFs leave, and you have the complete 70S initiation complex ready to go.
Okay, initiation complete. Now for elongation, adding the rest of the amino acids. Walk us through that cycle.
Right. Elongation is this repetitive cycle. First, the next charged tRNA, matching the codon now exposed in the A site, enters the A site. This is guided by an elongation factor called EF2, which uses GTP.
So a new tRNA comes into the A site, then what?
Then comes the critical step,
Yeah.
peptide bond formation. The amino acid, or growing chain, attached to the tRNA in the P site is linked to the amino acid on the tRNA in the A site. And remember that catalytic RNA in the large subunit?
The ribosome.
That's the one. The 23S RNA in bacteria catalyzes this peptide bond formation.
Incredible. Okay, bond formed. Now the chain is attached to the tRNA in the A site. How does the ribosome move to the next codon?
This is called translocation. The entire ribosome shifts exactly 3 nucleotides, 1 codon along the mRNA in the 5' to 3' direction. This movement shifts the tRNA that was in the P site, which is now in charge, to the E site where it exits. The tRNA that was in the A site, now carrying the growing polypeptide chain, moves into the P site. This leaves the A site empty, ready for the next charged tRNA to come in. This whole translocation step requires another elongation factor, EFG, M or GTP hydrolysis.
And the cycle just repeats, codon by codon. It sounds efficient.
It's remarkably efficient and fast. In E. coli, it can add something like 15 amino acids per second, and it's incredibly accurate, with an error rate around 1 in 10,000 amino acids. And cells make it even more efficient by having multiple ribosomes translate the same mRNA molecule simultaneously.
Yeah.
These structures are called polyribosomes or polysomes. You can actually see them under an electron microscope like beads on a string, with polypeptide chains getting longer at each bead.
Polysomes, that makes sense for mass production. Okay, so it elongates until when does it stop?
Termination. This happens when one of 3 specific codons, UAG, UAA, or UGA, enters the A site. These are the stop codons.
And there are no tRNAs for these.
Correct. No tRNAs recognize these codons. Instead, proteins called release factors, RS, recognize them. RF1 or RF2 binds to the stop codon in the A site.
And what does the release factor do?
Its binding triggers the hydrolysis of the bond connecting the completed polypeptide chain to the tRNA in the P site. The polypeptide is released. Then another factor, RF3, helps release the tRNA and causes the ribosome subunits to dissociate from the mRNA, ready to start again.
So stop codons and release factors signal the end.
Got it.
Now, you mentioned eukaryotes are different. What are the key distinctions in their translation process?
Several key things. First, the location. In eukaryotes, transcription, DNA to mRNA, happens in the nucleus, while translation, mRNA to protein, happens out in the cytoplasm. This separation allows for more regulatory steps.
Right, unlike bacteria where it's all coupled together.
Exactly. Second, eukaryotic ribosomes, 80S, are larger and more complex than bacterial ones, 70S. And third, initiation is quite different and generally more complex.
How so? No shine-dalgarno sequence.
No shine-dalgarno. Instead, eukaryotic initiation usually relies on the 5' cap that modified guanosine added to the start of eukaryotic mRNAs. The small ribosomal subunit, along with many eukaryotic initiation factors, EAFs, binds near the cap and then typically scans along the mRNA until it finds the first AUG codon.
It scans, like it slides along.
Sort of, yeah. It slides along looking for the start signal. And the initiator tRNA in eukaryotes carries regular methionine, not fmet, although it's still a special initiator tRNA. Often there's a sequence around the AUG called the Kozak sequence that helps increase initiation efficiency.
Interesting. And I've heard about closed-loop translation in eukaryotes. What's that about?
Ah, yes. That's a neat regulatory feature. It involves interactions between proteins bound to the 5' cap, like EIF4E, and proteins bound to the 3' poly-A tail, poly-A binding proteins, PAPs, often bridged by another factor like EIF4G.
So the beginning and end of the mRNA are linked.
Effectively, yes. It forms a loop. This is thought to increase translation efficiency, maybe by helping ribosomes that finish translating quickly reinitiate on the same mRNA. And it also might serve as a quality control check, ensuring only intact mRNAs are translated. Plus, eukaryotic mRNAs generally last much longer than bacterial ones.
That makes sense. More regulation, more stability. And what about that high-resolution structure of the human ribosome from 2015? Any key takeaways there?
That work by Kloholz and colleagues was fantastic. Seeing the human ADS ribosome at such detail confirmed things like the subunit rotations during translocation. Understanding these differences between bacterial and human ribosomes is absolutely vital for developing better antibiotics, drugs that target bacterial ribosomes but leave ours alone.
Right. Specificity is key.
And potentially, it could help in designing cancer drugs, maybe ones that selectively slow down the hyperactive ribosomes in tumor cells.
It really highlights how fundamental research translates into practical applications. Okay, let's shift gears a bit and look back. How did scientists originally figure out that proteins were the direct products of genes? It wasn't always obvious, was it?
Not at all. One of the earliest clues came from Sir Archibald Garrod, a physician back in the early 1900s. He studied inherited human disorders.
Like alkaptonuria.
Exactly, alkaptonuria, where people have black urine because they can't break down a certain acid. Gary noticed it ran in families, followed inheritance patterns, and proposed it was due to a missing step in a metabolic pathway, essentially, a faulty ferment, or what we now call an enzyme. He called these inborn errors of metabolism.
So he linked heredity to specific biochemical reactions very early on.
He did, though his work wasn't fully appreciated at the time. It was a remarkable insight, linking genes, or unit factors as they called them then, to enzymes.
Then came Beadle and Tatum in the 1940s with their famous experiments on bread mold.
Right, George Beadle and Edward Tatum, working with Neurospora crassa, they deliberately created mutations using X-rays and then looked for mutants that couldn't grow unless they were given specific nutrients.
Auxotrophs.
Yes, auxotrophs. By finding mutants that needed, say, a specific vitamin or amino acid, they could deduce that the mutation had knocked out the enzyme responsible for making that nutrient. This led them to propose the one gene, one enzyme hypothesis.
One gene codes for one enzyme, a huge conceptual leap.
Absolutely. It provided strong experimental backing for the idea that genes control biochemistry by specifying enzymes.
But that hypothesis needed refining, didn't it? Because not all proteins are enzymes, and some proteins have multiple parts.
Correct. It evolved into the one gene, one polypeptide chain hypothesis. And the classic evidence for this came from studying sickle cell anemia.
Ah, sickle cell. How did that provide the proof?
Well, sickle cell anemia is clearly inherited. It affects hemoglobin, which is a protein but not an enzyme. In 1949, Linus Pauling showed that hemoglobin from people with sickle cell disease, HBS, behaved differently in an electric field than normal hemoglobin, HbA, indicating a chemical difference.
Okay, so a chemical difference in the protein linked to the inherited disease.
Exactly. And then Vernon Ingram, a few years later, did the painstaking work of figuring out exactly what that difference was. He found it was just a single amino acid change in the beta-globin polypeptide chain.
Just one amino acid?
Just one. At the 6th position, normal hemoglobin has glutamic acid, while sickle cell hemoglobin has valine instead. A single point mutation in the gene led to a single amino acid change in the polypeptide.
Wow. That's incredibly specific. What was the impact of that discovery?
It was profound. It definitively confirmed the one gene, one polypeptide idea, showed how a tiny change in a gene could have major physiological consequences and really established the whole field of molecular medicine understanding diseases at the level of protein structure.
Okay, so we've established that genes code for polypeptide chains, but a linear chain isn't usually functional, right? That brings us to protein structure. What's the key difference between a polypeptide and a protein?
You're right. Polypeptide is the linear sequence of amino acids linked by peptide bonds. A protein is that polypeptide chain, or multiple chains, folded into a specific, stable, three-dimensional shape that allows it to perform its function.
And the building blocks are the 20 common amino acids, each with a different side chain, or R group.
Exactly.
Yeah.
And the huge variety in these R groups, nonpolar, polar charged, is what gives proteins their incredible chemical diversity and allows them to fold in so many ways and perform so many functions. The number of possible sequences is just astronomical.
And we talk about 4 levels of protein structure. Can you quickly outline those?
Sure. First is the primary structure, simply the linear sequence of amino acids. This is directly dictated by the gene via mRNA.
Okay, the basic sequence.
Second is the secondary structure. These are local repeating folding patterns, mainly the alpha helix, a spiral, and the beta-pleated sheet, like folded paper. These are stabilized by hydrogen bonds along the polypeptide backbone.
Alpha helix and beta sheet. Got it. What comes next?
Tertiary structure. This is the overall three-dimensional shape of a single folded polypeptide chain. It's determined by interactions between the R groups, hydrophobic interactions, hydrogen bonds, ionic bonds, disulfide bridges. This unique 3D shape is essential for the protein's function.
So the final fold of one chain and the fourth level.
Quaternary structure only applies to proteins made of more than one polypeptide chain, or subunit. It describes how these multiple subunits fit together and interact to form the final functional protein complex, like hemoglobin, which has 4 subunits.
Okay, primary, secondary, tertiary, and sometimes quaternary structure. Now, folding sounds complicated. How does the cell get it right, and what happens when it goes wrong?
It is complicated. Many proteins can actually fold spontaneously into their correct shape just based on their amino acid sequence. The interactions guide it to the lowest energy state. But others need help.
Help from what?
From other proteins called chaperones or chaperonins. These bind to unfolded or partially folded polypeptides and help guide them along the correct folding pathway, preventing them from sticking together incorrectly or getting stuck in non-functional shapes.
So, molecular chaperones, what if folding still goes wrong?
The cell has quality control systems. Misfolded proteins can often be recognized, tagged with a small protein called ubiquitin, and then targeted for destruction by a complex called a proteasome.
Like cellular garbage disposal for bad proteins?
Sort of, yes.
Yeah.
But if misfolded proteins accumulate, either because the system is overwhelmed or the proteins are resistant to degradation, they can form aggregates that are toxic to cells.
And this leads to disease.
It certainly can. Prion diseases, like Creutzfeldt-Jakob disease or mad cow disease, are a dramatic example. Here, a misfolded form of a normal brain protein, PRP, somehow induces other normal PRP molecules to misfold in the same way, leading to devastating aggregation and brain damage. It's essentially a disease of protein secondary structure propagation.
That's terrifying. A misfolded protein causing others to misfold. Are other diseases linked to this?
Yes, many. Sickle cell anemia involves the aggregation of the mutated hemoglobin, and several major neurodegenerative diseases, including Alzheimer's, Parkinson's, and Huntington's, are characterized by the accumulation of specific misfolded protein aggregates in brain cells. Protein folding and misfolding is a huge area of research.
It sounds incredibly important. So putting it all together, while DNA holds the blueprint, proteins are really doing the work in defining the diversity of life. Just how diverse are their roles?
Immensely diverse. Proteins are arguably the most versatile macromolecules. They transport molecules like hemoglobin carrying oxygen. They provide structure, like collagen in our connective tissues or keratin in hair. They enable movement, like actin and myosin in muscles. They defend the body as antibodies. They regulate processes as hormones or transcription factors, controlling which genes are turned on or off.
And enzymes, of course.
And enzymes, yes. Probably the largest and most diverse group. They act as biological catalysts, speeding up virtually every chemical reaction in the cell with incredible specificity. A cell's entire metabolism depends on its suite of enzymes.
And within these complex protein structures, there are often distinct regions called domains. What are they?
Right. Protein domains are distinct structural and functional units within a larger polypeptide chain. Think of them as modules, often 50 to 300 amino acids long, that fold up somewhat independently.
And each domain might do something specific.
Exactly. A protein might have, say, a domain that binds DNA, another domain that binds a signaling molecule, and a third domain that has enzymatic activity. This modular nature allows for great versatility. Evolution can mix and match domains to create proteins with new combinations of functions.
That's a really neat concept, proteins built from functional modules. Well, we've covered a lot of ground today. We've journeyed all the way from the mRNA code to the complex, folded, functional proteins that are the true workhorses of the cell. The precision and intricacy are just staggering.
It truly is. We hope this deep dive has helped clarify this fundamental process for you, connecting the dots from the genetic code to the ribosome's function, the tRNA's role, and how we even discovered these things historically is full of amazing molecular details.
And as a final thought to leave you with, considering how critical correct protein folding is and how devastating misfolding can be, what might the future hold? Could we develop ways to routinely correct protein misfolding errors that cause diseases like Alzheimer's or Parkinson's? Or even design completely novel proteins to perform specific tasks in medicine or industry? The potential seems enormous.
It really does. The more we understand these fundamental processes, the more possibilities open up. Thank you for joining us on this deep dive into translation of proteins. Till next time, keep exploring the wonders of the molecular world.